In this presentation, I am going to talk about the forensic analysis of alpha-amylase in saliva. Saliva is an extracellular biological fluid secreted by salivary glands in the mouth. Saliva in human consists of water, electrolytes, mucus, white blood cells, epithelial cells, amylase, lipase, immunoglobulin A, and lysozymes. Alpha-amylase is present in human, elephants, rats, and pigs. Saliva can be found in cigarette butts, clothes, bottle cans, handkerchief for forensic analysis. Starch iodine and Fodebas test are the presumptive tests of saliva, and rapid stain identification test is a confirmatory test used to detect saliva. Starch iodine presumptive test. Enzyme amylase is a component of saliva. To the sample, 0.5% soluble starch solution and glucose iodine solution should be added. If iodine and starch bind to form a starchy iodine purplish complex, then the amylase is not present. If the enzyme hydrolyzes starch, turning the sample colorless, then it indicates that salivary amylase is present in the sample. Amylase is also present in other biological materials such as feces, vaginal fluids, sweat, and also in urine. The next presumptive test is Fodebas test. Fodebas amylase test was developed by Pharmacia Diagnostics in the year 1970. Fodebas is a biochemical substrate. It is used for the detection of alpha-amylase enzyme. The name Fodebas is an abbreviation of Pharmacia Diagnostics biologically active substance. So this Fodebas is a tablet that is water insoluble starch cross-linked to blue dye, which get hydrolyzes to blue fragments in the presence of alpha-amylase. So the active component in this Fodebas tablet is DSM-P microspheres, in which a blue dye has been bound. When the substrate is digested by the enzyme present in the solution, the color turns blue, indicating that alpha-amylase is present. So the Fodebas forensic press test is used for identifying saliva stains. Rapid stain identification test for saliva. Rapid stain identification test is also known as lateral flow immunochromatographic strip confirmatory test for the detection of human salivary enzyme alpha-amylase. The RSID confirmatory test uses two mouse monoclonal antibodies specific for salivary alpha-amylase. One of the antibody is conjugated to colloidal gold and it is deposited beneath the sample window. The other antibody is strapped onto the test line on a membrane attached to a conjugate pad. The other end of the membrane is a wick that absorbs the tested fluid and the running buffer after the completion of the test. When the sample is added to the sample window, the running buffer and the sample diffuse through the conjugate pad, redissolving the gold conjugated detection antibodies. If human salivary amylase is present in the sample, antigen colloidal gold conjugated antibody complex will be formed and a red line will be produced both in the test line and also in the control zone. If human salivary enzyme is not present in the sample, then antigen antibody complex will not be formed and a red line will appear only in the control zone. The diagram is shown here.